Good afternoon. Welcome to the NINDS IGNITE webinar. I'm Becky Roof. I manage the IGNITE program, and IGNITE stands for Innovation Grants to Nurture Initial Translational Efforts.
Hello, I'm Chardell Spriggs, the Program Officer with the IGNITE program.
Hi, I'm Julia Bachman. I'm a Health Program Specialist with the IGNITE program.
And I am Tim Leiden. I'm the Operations Coordinator with the IGNITE program. The way that this meeting or webinar is going to work is Dr. Roof, Dr. Spriggs, and Dr. Bachman will be giving a brief presentation on the program. And then afterwards, you will be given the opportunity to ask questions. You're wondering, how can I ask questions if I'm muted? The way that you do it is through the Q&A field. Now, if you hover over with your mouse in your WebEx, if you see that dot, you just click on it, and you'll see Q&A, and that is where you ask the question. Please only use the chat if you have an issue with your audio or if you can't see something, but all questions, please use the Q&A field. Lastly, this meeting is going to be recorded. The hope is that we will be able to send this recording out to everybody that is registered. At the very least, we will definitely be sending you out the slides this week. So with that, I will turn it over to Dr. Roof, and we'll start the show.
Great. Thanks, Tim. Also, Ashley, did you want to introduce yourself briefly?
I'm sorry. Hello, my name is Ashley Gibbons, and I'm also the Operations Coordinator with the IGNITE program.
Great. Thank you, Ashley. All right. So let's get started. So the IGNITE program is a early stage therapy development program. It's really designed to help people get what they need to be ready for later stage programs, such as the Create Bio program for biologics, or the NIH Blueprint Neurotherapeutics program for small molecules. Once you receive these slides, these links will be live, and you'll be able to click on them for more information on both of these programs. So IGNITE, as I said, is an early stage translational program. It has 3 funding opportunity announcements, one for therapeutic agent characterization and in vivo efficacy studies, one for assay development and therapeutic agent identification, and one for the development and validation of animal models as well as PD markers. We will talk about each of these in more detail later on. But first, the general overview, they are 3-year programs with a maximum budget of $750,000 direct costs for the entire project, with no more than $499 in any given year. I'll also point out that the next 2 receipt dates are February 19th and June 17th. There are 2 additional receipt dates as well. Please notice that the resubmission, A1 resubmission date is the same as the new application date. Please don't email me on February 20th saying I still have a month, right? No, it's the same date for everybody. So please pay attention to that. I will point out that if you are eligible for continuous submission, or if you have some family emergency or something, this notice here might apply to you. But please note that, and that notice says that late submissions up to 2 weeks can be submitted in certain circumstances. So you can take a look at that, but please note that it's not a guarantee, and I can't guarantee anything in advance. So it's really best to submit on time. In fact, it's best to submit early in case there are issues. All right, so I'm going to spend a minute and talk a little bit about this mechanism, since it is different for a lot of folks. It is not the same as your regular R01 or R21 type mechanism. It's 2 phases. There's an R61 phase where you can demonstrate feasibility, get ready, get your ducks in a row. And then there's the second phase where you do the main event. So, for example, you might use the first phase to develop assays and the second phase to do a high throughput screen, or you might use the first phase to optimize a compound and then the second phase to do your in vivo proof of concept efficacy study. So it's really important to note, though, that there's this milestone gate in between the 2 mechanisms, or between the 2 phases. So let me step back and talk a little bit more about the milestone. So this is something that a lot of people have trouble with. It's different compared to regular R01s and R21s. And a lot of people wonder, why would you even do a mechanism that has a milestone in it, it really allows you to propose riskier science, where the second phase depends on the first phase working. And that's something that's hard to get funded in other mechanisms, especially if the second phase is expensive and the first phase is risky. But dependent aims are really expected in this mechanism. And the reason that that works is because of this milestone. This milestone gives reviewers confidence that, you're only going to do the second phase if the first phase works. And that's why, we're okay with dependent aims. So in order for that to be successful, the milestones need to be clear and quantitative, and really support, the idea that the second phase should be done because the first phase was successful. You've demonstrated that it's feasible. You've demonstrated that you've prepared. Your assay is ready for the high throughput screen, or your compound is ready for in vivo testing. So, think about the milestone not as a barrier, but as a, checkpoint that, it's worth going forward to the second phase. I'll also point out that there's, a single go, no-go transition point between the two phases. That's different for some of the other translational programs where there's multiple go, no-go decision points. And also point out that the R33 transition is an administrative review, which means it's done internally at NIH. It doesn't go out to peer review again. If everything is smooth and your paperwork's in order and we all agree your milestone's met, you can go from your R61 to the R33 without any delay or lapse in funding. So because the milestones are, new and different for a lot of people, we put some examples in here. I'll also point out that we have examples on our website if you want to see more. But, an example of a weak milestone that often doesn't do well in review would be, for example, scale up of a compound. It shouldn't be a list of activities that you're going to do. It really needs to be what you need to see in order to go forward. So a stronger milestone might be, we'll demonstrate, we'll generate so many grams of it, there's going to be a certain amount of purity, for example. And again, these are all examples. These are not, we don't dictate milestones here, but a weak milestone example might be we would perform PK studies, just a list of what activities you're going to do. A stronger milestone would be something like, these are the properties that we need to see in the compound, half-life, the brain-plasma ratio, et cetera. A weak milestone might be, we'll fully optimize our assay. A stronger milestone defines what that looks like. What a Z-score should be, or a signal to noise, whatever's appropriate for your particular situation. Another weak milestone might be, we're going to see a decrease in seizures. A stronger milestone would be we're going to see a decrease by X% for Y duration, whatever. And these are just examples. I don't want to, we don't dictate anything. But it's really important that it be clear and quantitative. There shouldn't be any fuzziness. It shouldn't be any question when the time comes whether the milestone's met or not. It's also really helpful, if, for example, in the last one, to talk about where X came from. So if you need to see a 20% decrease in seizures, well, why is 20% expected to be clinically meaningful, for example? Okay, so now we're going to switch. Oops, I'm sorry. Again, the milestone examples are on the website. And again, this link will be live when you get the actual slides. So we're going to switch over now and talk about each of the FOAs individually. So Chardell, would you like to start out?
Sure. The PAR 18761 is the pharmacodynamics of the neurotherapeutic agent characterization and in vivo efficacy studies funding opportunity announcement, or FOA. This FOA provides funding for studies to demonstrate that early-stage neurotherapeutics have sufficient biological activity to warrant further development. We use a combination of parameters, including target engagement, pharmacodynamics, pharmacokinetics, and in vivo efficacy studies to determine the feasibility of a candidate therapeutic agent. There are some entry criteria that are necessary, and you should be sure to illustrate them in your application. They are novelty, biological rationale, and relevance for therapy development. This FOA seeks to apply new knowledge around targets, mechanisms, pathways, or therapeutic indications. Projects should aim to develop or repurpose therapeutics that are significant improvements over existing therapeutics to treat neurological disorders. We like to see a strong biological rationale for the intended approach, along with preliminary data or literature-based evidence. supporting the feasibility and importance of the approach, including evidence that a therapeutic agent has the potential to be therapeutically viable, as well as evidence to support the robustness of the pharmacodynamic measures and/or in vivo efficacy models. Preliminary data should be from well-designed experiments. Projects should include a description of the unmet need for the therapeutic agents and a clear justification for how the findings from these studies are relevant to treatments for disorders that are within the NINDS mission. Projects should also explain how the candidate therapeutics will be optimized and advanced toward later stages of discovery once the R33 phase is complete. Examples of activities for the R61 phase include, but are not limited to, some of these we have listed here. Preparation of the therapeutic agent to support proposed activities, characterization of the therapeutic agent. This could be confirmation of purity, stability, ADME, in vivo potency and selectivity, degradation products, and process impurities. Focus studies to optimize the candidate therapeutic agents for in vivo efficacy testing, or those to develop dosage forms to support proposed PD measures and/or in vivo studies. Pharmacokinetic studies to determine therapeutic agent levels. There are studies to confirm that therapeutic agents reach and engage the target site, or studies to inform design and refinement of the PD measure and/or in vivo efficacy models and testing procedures, including examination of model variability to estimate sample size, calibration of the model according to positive and negative controls, along with the age, time course, and window of disease that are relevant for therapeutic testing. At the end of the R61 phase, investigators must exhibit successful completion of all necessary preparation and characterization of proposed therapeutic agents, such as chemical synthesis and scale-up to provide material for proposed projects, in vitro or in vivo potency studies. They should have all pharmacokinetic studies concluded, the design, refinement, and validation with pharmacodynamic markers using the appropriate positive and negative controls again, and demonstrating feasibility of conducting the therapeutic agent testing, as well as a further refined or detailed in vivo study design that meets the NINDS rigor guidelines and will allow for demonstration of dose and exposure response. Examples of activities for the R33 phase include, but are not limited to those listed here. They include pharmacodynamic and/or in vivo efficacy studies with chemically and biologically characterized therapeutic agents, dose response activity with the intended route of administration, studies correlating pharmacokinetic and pharmacodynamic measures, and validation and replication studies to confirm observed results. There are some activities that are either not in scope of the IGNITE program or do not specifically fall within this funding opportunity announcement, but may be met with another funding opportunity announcement within IGNITE. Examples of some of these activities that are not appropriate for this FOA include, but are not limited to, study or use of therapeutic agents to identify targets relevant to a disease, studies of disease mechanisms, development of de novo animal models and pharmacodynamic measures. These are covered under the PAR 18763. Assay development, identification of novel therapeutic agents, and these will be covered later under PAR 18762. GLP toxicology studies, IND-enabling studies with optimized therapeutic agents are also out-of-scope activities. Discovery or development of devices, surgical procedures, diagnostics, or rehabilitation strategies. Discovery and development of risk, detection, diagnostic, prognostic, predictive, or any biomarker are also out-of-scope activities, as well as manufacture of therapeutic agents for clinical trials. There are some exceptions of clinical research and clinical trials, which Dr. Bachman will go over later on within this webinar.
All right, thank you. So this is Becky Roof again. I'm going to talk about PAR 18762, which is for assay development and therapeutic identification and characterization. So the goals of this program are to develop new assays and also to screen to identify and characterize novel therapeutic agents. Now, a lot of people use this mechanism for high-throughput screening, but you don't have to do high-throughput screening with this. There are other things that would also be appropriate. And if you are interested in talking about a particular example, I'm happy to discuss that. The entry criteria for this are going to look very familiar. They're a lot like what Chardell just talked about. Their novelty, strong rationale, relevance for therapy development, and obviously having availability, test agents or library if you're hoping to do any kind of screening or characterization. Now, if you're doing like a computational screen or something, you wouldn't obviously have those test agents, so that's okay. So for the R61 phase, some examples include, but are not limited to, things like developing assays. Obviously, that's one of the goals of the program. You might use this for developing your primary assay or your secondary assays or your selectivity assays or whatever else you need to support your testing funnel. Some people sometimes have questions about phenotypic assays, and that's quite fine. There's really no concern around that, I don't think. You can use it for developing in vitro or ex vivo assays. You can use the first phase for developing assays that you will later use to evaluate properties. If you're developing some kind of protein and you want to look at aggregation, or you're developing some kind of virus and you want to look at infection, for example, anything that you would need to later use to evaluate the properties, you can develop assays for. You can use it to develop assays to look at purity or identity of hits that you would find later on. Any kind of assay development that you might need, for example, for high-throughput screening, miniaturization, testing, your Z-score, whatever else. And of course, you can use a combination of assays if that's appropriate for your testing funnel.
Use your mouse to go over and click on it.
Sorry about that. All right. So by the end of the R61, some examples of things that you might have hoped to have accomplished might be to have your assay developed and optimized, to have that complete. You might need to do physiochemical or biophysical characterization of compounds that you are going to test in your second phase. Anything that you need to develop to, later look at selection of cell lines or vectors, or depending on, whatever it is you're developing, anything that you would need to validate your hits later on. For the R33, you might do some of the following activities. It's not limited to the below. You might do your high-throughput screening. You might optimize compounds. You might prepare your therapeutic agents, confirm structure, assess properties using assays you developed in the first phase. You might begin to do some early-stage ADME. You might look at selectivity of your hits. These are all examples of things you might do in the R33. Again, there are some out-of-scope activities. Some of these are repeats from the last PAR, and they fall into categories of things that are either supported through other mechanisms or are earlier stage or later stage than what this supports. So development of assays or probes to support basic understanding of disease. We don't support basic science with this. This is a translational focused mechanism. Pharmacodynamic and in vivo efficacy studies are supported by PAR 18761 that Chardell just talked about. Devices are supported by another program. If you're interested in device development, I can point you to that program. Target identification is generally considered more basic science. Although if you did some phenotypic screen and then you need to do a little bit of target identification, we can talk about that. Development of biomarkers, we have a whole separate biomarker program. So if you're looking to do biomarkers, you're better off over there. IND-enabling studies and clinical studies tend to be later stage and would not be appropriate for IGNITE. And again, clinical research and clinical trials but we'll hear more about that later.
Okay, so this is Julia Bachman. I'm going to talk about the third funding opportunity in IGNITE, which is PAR 18763. And this is for the development and validation of model systems and pharmacodynamic markers. The goals of this funding opportunity are, one, to promote a significant improvement in the translational relevance of animal models, ex vivo systems, and pharmacodynamic markers that will be utilized to facilitate the development of neurotherapeutics. So essentially, we're looking to provide translational tools for the drug development process. And not just for you and your lab, but something that would really make a difference for the whole disease area in the field, and something that other investigators or companies would want to use as they're developing candidate neurotherapeutics. Another goal is to develop a fully validated model system, testing paradigm, or pharmacodynamic marker that can be used in both preclinical and clinical settings to test the biological effects of a candidate neurotherapeutic agent. The emphasis here is that we want a very rigorous approach that is going to be translational, i.e. the model will mimic as much as possible the human disease or condition, and the endpoints that are used to address that will be endpoints that, for the most part, can be used in a clinical setting. So overall, in addition to having a strong biological rationale and translational rationale for your model or pharmacodynamic marker, there should be evidence of value for future drug discovery and development. Okay, to go over some relevant definitions for the context of this funding opportunity. First, a pharmacodynamic marker is a specific type of biomarker whose level changes in response to a therapeutic intervention. This is typically a component of the molecular pathway mediating the biological effects of a therapeutic target modulation, or a component of disease etiology that is involved in drug target modulation, and this can be either direct or indirect. Internal validation addresses the consistency of the experimental design. It involves characterizing the precision, reliability, analytical sensitivity, accuracy, and dynamic range of the endpoints utilized in the model system or biomarker measurements, whereas external validation refers to the extent to which studies or results of a study can be generalized from one setting to another. And there are typically three different types of external validation. Face validity refers to the similarity between a model or model system and clinical manifestations of the disease, so in terms of the symptoms, morphology, and so forth. Construct validity is the similarity between model or model system and the physiological basis of the human disorder in terms of what is known about the disease mechanism. And finally, predictive validity is the similarity between the effect of a validated therapeutic intervention in the model or model system and the clinical disease population. Example activities for the R61 phase of this funding opportunity. Sorry. Oh, there we go. Okay, so example activities include the initial development of the model, ex vivo system, or pharmacodynamic marker. And you can completely develop a model from scratch, or you can take a model that hasn't been well validated and validate it. So you want to include any optimization related to the feasibility, endpoint range, sensitivity, and those types of measures, so the internal validation for the endpoints used in these model systems or the marker, as well as any scale-up needed for the R33 phase. In particular, for example, would be if you're developing a genetically modified animal. Will the breeding be sufficient so that in the end you'll have enough animals from a stable generation that you can use for the drug testing or the necessary external validation studies? And by the end of the R61 phase, you want to be able to show that your model system or pharmacodynamic marker is feasible for external validation studies. So this can be just the actual ends for your animals or the basic internal validation studies for your endpoints. You also want to show that it reflects the functional or physiological pathway of the disease or therapeutic target as intended. And again, your milestone should address these aspects in a quantitative way. Examples of activities for the R33 phase include all of the external validation studies, including comparisons of the model phenotype to human disease, comparisons of disease etiology in preclinical species to what is known about the human disease, or comparing the efficacy of clinically validated therapeutic agents in the new model system if such therapeutics are available. Okay, activities that are out of scope for this funding opportunity include the development of animal or ex vivo cellular models for the purpose of understanding disease etiology. So this would be more appropriate for an exploratory R21 or regular R01 mechanism. Cell line development and the identification of CNS drug targets is also out of scope. the discovery and development of disease initiation, remission, relapse, or progression biomarkers. So again, this funding opportunity only supports the development of pharmacodynamic markers, but as Becky mentioned previously, we have a biomarker program, and we're actually having a webinar for that program next Wednesday for those who are interested in learning more about those funding opportunities. Other out-of-scope activities would be those that fall under one of the other two IGNITE FOAs that we've previously discussed, so I won't go through them in more detail here. Device discovery and or development. So again, NINDS has a separate program for devices, and we've included the link here for that program if you're interested. And finally, clinical research and clinical trials, which we'll go through shortly.
All right. So I'd like to leave you with some general tips, and these apply to all three FOAs. You can contact us in advance. We're happy to look at your AIMS page, including milestones, and talk to you about how this mechanism can be used, how your particular project might fit, how to work with phases and milestones, and all of the things that are unique to this program. We can also talk to you about things that reviewers could potentially question or have issues with. We've seen a lot of reviews, and there are some common repeat issues, and we can help you try to avoid those. I've already talked about milestones, but I'll mention it one more time because this is something that people struggle with. They need to be clear, and we have examples on our website, and we're happy to talk to you about your particular examples if you'd like to talk to us. You've heard before rigorous study design. NINDS has been promoting rigor and reproducibility for a few years now. This is always important, but it's particularly important here. Reviewers are asked to look at rigor and reproducibility and comment on it. So this is a really easy thing to avoid having a problem around because you know it's going to come. Have a multidisciplinary team. So having deep disease biology expertise is not enough here. You need to also include members of your team who know translation. You know, you maybe have clinicians who know the disease and who can speak to the fact that you're developing something that's meaningful. Depending on your project, you might need chemists, you might need assay biologists, you might need animal model experts, whatever is appropriate for your study, but have a multidisciplinary team. And also pay attention to the fact that the review will be multidisciplinary. So this review is done by NINDS. You will have 4 or 5 reviewers. And we do that so that we get 4 or 5 different experts with different perspectives. So you will have reviewers who know your disease, but also a chemist and also a translational person and whatever else is appropriate. So be aware of that. This is a very translationally focused mechanism with a very translationally focused review group. Many of our reviewers are from industry, so you're going to get that perspective on your review. Discuss intellectual property. This does not mean that you have to have a patent filed. In fact, in some cases, that's not what you would want to do at this stage. But it does mean that you have a plan. It does mean that you've talked to your tech transfer office. It's good to have a letter of support from them saying that they will work with you. Give reviewers confidence that there's some path forward. You just need freedom to operate at this point and some kind of plan. You also need to have a therapy development plan. Your multidisciplinary team can help you with this. Give reviewers confidence that this isn't, just an academic exercise, that at the end of the 3 years, you're going to have something, if you're successful, that has a potential path. So you might talk about, are there other treatments for this patient population out there? You might talk about what steps you would do after this grant if it's successful, what longer-term steps you might need to do, what kind of barriers you might anticipate, and give reviewers confidence that you've thought about it and that you have a way to overcome any issues. So that's also unique to this program, so it's something to think carefully about. If you are a small business, you are eligible for IGNITE, but I would highly recommend that you consider the SBI or STTR program instead. Congress mandates that we set aside funds for small businesses, and if you are a small business and you can compete with just other small businesses for those funds, you're in a much better shape. I would recommend you consider that path if you are eligible, and Emily can help you if you're not sure about your eligibility or if you want to consider that option. I will say that everything that we've talked about today in these 3 FOAs can be covered by the SBIR or STTR General Omnibus. So that is definitely something I would recommend if you're a small business. And lastly, pay attention to clinical research rules, and Jules will tell you more about that.
Yeah, now we wanted to briefly touch upon the NIH terms for human subjects and clinical research, since this can be a point of confusion for some of our applicants, especially when it comes to using human samples or patient-derived specimens, which have recently changed with the revised Common Rule. So NIH defines human subjects research as research involving a living individual about whom data or biospecimens are obtained, used, studied, or analyzed through an interaction or intervention, or where identifiable private information is used, studied, analyzed, or generated. Clinical research is defined by the NIH as research conducted with human subjects or on material of human origin, such as tissues and specimens, unless these studies fall under the CFR regulations for Exemption 4. So the federal regulations include 8 different types of exemptions, and we're specifically focused on Exemption 4. So what is Exemption 4? It involves the collection or study of data or specimens if the sources are publicly available or the information is recorded such that subject cannot be identified. Now the key is that Exemption 4 is for secondary research only. If the biospecimens were or will be collected specifically for the research proposed in the grant through an interaction with living individuals, then it's considered primary research. So overall, depending on the specifics of how your samples are collected and how they're coded or de-identified will determine whether it qualifies as human subjects research at all and or whether it would meet the Exemption 4 criteria. So the NIH's Office of Extramural Research provides a number of different great resources on their website, which we've included the link here. If you can go to the next slide. So this is what that page looks like, and you'll see there are a number of different resources with a lot of information, as well as FAQs and links to the actual guidelines and so forth. So we particularly like this decision tool. So if you go to the next slide. So it's a series of questions that you'll answer based on what your study involves. And after 3 or 4 questions, it'll let you know whether or not your studies are considered human subjects research and whether or not they're likely to qualify for Exemption 4. So it's the next slide. So we highly recommend that you look at the resources on this website, as well as, if you're not sure, talk to someone at your institute. So if your institute has an IRB or a designated human subjects officer, they would be a great person to go to. And you can also email OER directly. So we've included their email link specifically for human subjects related questions. So it's really good to figure this out before applying. And last but not least, you're welcome to email us, although we're likely to forward that email to OER to make sure. Okay.
Okay. Thank you, Julia. Yeah, it's really important to not assume on this one, because we've had a number of people who think they're not doing human research, and then it turns out they are, and you don't want to find that out late in the game. Okay, so thank you, everyone. If you have questions after this webinar is completed, you can contact Tim Lydon, and his information is here. You can also continue to send Q&A questions here through the webinar. If you have questions about a specific project, you can contact Tim or me or Julia or Chardell after the webinar, and we're happy to talk about a particular project. And lastly, you can find more information on our website, including those milestone examples, and you'll be getting all of these slides by email if you've registered.
Okay, so let's go on to our first question. Can foreign extramural scientists ask for a grant or be awarded for an exceptional scientific translational research?
Hi, that's a great question. So yes, you are eligible to apply, but I'll point out that the bar for award is a little bit higher. So if you are a foreign researcher, you will have to get approval, not just the regular, you got a great score, but you will also need to have approval specifically by our council. And in order to get that, you need to make the case that this work couldn't be done domestically. And so that's actually quite a high bar, and it's a little bit of a challenge. But you are eligible, and if you want to pursue that, please contact me in advance, and we'll talk about it.
Okay, next question. For funding opportunity PAR 18762, do we have to show the structures of a hit compounds in this application?
So, so that would be the high throughput, or the, not necessarily high throughput screen, but the screening and agent identification FOA. Right, so you wouldn't have your hits prior to doing the grant, right? You would have it would come up during the grant award. And we generally would like to see structures at the end of the grant to show that what you've accomplished. And anytime we do final progress reports, we like to know how things turned out. But that is, if it's a final progress report, that's only shared with NIH. I hope that was the right question. If you're talking about PAR 18761, which would be the in vivo efficacy studies, and you have a compound in hand coming in that case, it's not required, but it's strongly recommended. Reviewers will want to see the chemical structure. We have chemists on our review group, and they'll want to critique that, whether or not they think it's a viable drug, in order to give you a good review.
Next question. How can you address that some reviewers focusing on applicants' language and publication instead of science?
So grantsmanship issues do sometimes come up. It sounds like you have a particular example in mind, and it would really be helpful to talk specifics about your particular summary statement. And I'm happy to do that if you want to contact me about it. This doesn't sound like it's a general question. It sounds like somebody's got a particular concern. But in general, grantsmanship issues do sometimes matter if it makes it hard for the reviewers to understand what you're talking about. But contact me and we'll discuss specifics.
Next question. Would an organoid ex vivo model fit the PAR 18763?
Yeah, we do support ex vivo. We honestly don't get a lot of those applications, but it does support that, and we can talk about it if you want to send me your specific information.
Okay, next question. What if the technology has been licensed by a company already? Can the tech still be investigated through the academic lab? If so, are there special requirements? What if only the lead is licensed, but there may be related compounds that are worthy?
Yeah, that's totally appropriate. So, that just speaks to your therapy development plan and your intellectual property path already being, underway. I would think that would be a good thing. We would just have to make sure that there isn't any overlap between what the company is supporting you to do and what you're asking for in the grant.
Okay, next question. For PAR 18762, do I need to contact HTS facility before applying?
Yeah, it would be good to have your partnership lined up for the high throughput screen, because that's something, the facilities and the environment are something that reviews critique, right? So they want to know who that is. There are a number of facilities available, and even NIH NCATS has a facility, but I would encourage you to have that collaboration lined up in advance.
Next question is what each of the PAR numbers is and is not looking for. And examples would be very helpful. Platforms, ex vivo versus human on chip definitions, and off-the-shelf drug models. So we hope we've covered a lot of this during the presentation today. I think the best thing to do is to contact us with your specific aim page and tell us what you want to do. And then our job is to find the best funding opportunity for you, whether that be in IGNITE or through a standard R01 still in NINDS.
Okay, next question. Which entity has a better chance of getting funded, a small company or an academic lab?
All right, well, so if you're applying to IGNITE, we don't make a preference between academic labs or small businesses. right? If you are a small business, though, as I said, you can apply through the small business program, and that, I think, is a better chance just because of you're only competing with other small businesses.
Next question. How much preliminary data with test or lead compounds in an in vivo model are required for strong application?
Yeah, so it's hard to make a blanket statement here. So it really depends on your project. You know, you really want to speak to those entry criteria that we talked about. You know, and that can be with your own preliminary data or with stuff from the literature. So you really want to demonstrate, you know, relevance for therapy development and novelty, all of these things that we talked about, and that can be preliminary data or, you know, literature support. It's really hard for me to make a blanket statement about how much preliminary data is needed.
Next question. Would products related to stem cells and stem cell-derived extracellular vesicles be eligible for these calls?
Yes. You're probably talking about the in vivo efficacy one here, I'm guessing. Now, with all of the regular rules and regulations around stem cells and stem cell research, but yes, cell therapies are appropriate here.
Can K awardees apply for IGNITE? How does ESI status affect the IGNITE award decisions?
Okay, so for the ESI status, you do not get a special bonus for being an ESI or an early stage or a K awardee in this mechanism, because it's not one of those mechanisms where that helps you. I will point out that if you have ESI status, getting the R61 does not impact your status for future R01 applications, but getting the R33 would. Now, if you're really worried about that, there are things you can do. You can delay the R33. You can ask for up to 3 years in the R61. You can even choose to not accept the R33 if you're really worried about your status. But to answer the question, no, the ESI status doesn't help you at all here.
If you have strong preliminary data, do you need to come up with a milestone for the transition from R61 to R33 phase that is truly go, no-go?
Yes, you do. You always need a milestone. Now, some people will come in saying the R61 stuff is already done and they want to go straight to the R33. We can't do that with these FOAs. It's just not within, you know, our ability to do. But you can play around with things. So, you know, we recommend and hear what things you might do in the R33 versus the R61. But if your R61 is already done, say you already have your assays developed for a high throughput screen, you could do a high throughput screen in your R61 phase and then do optimization of your compound in the R33, for example. So we can play around with the phases a little bit, but you do still need to have a milestone.
Next question. What happens if there is need for drug synthesis, but the synthesis is very expensive and interferes with the ability to carry out the work?
Yeah, so unfortunately we do have these budget caps and we really can't do anything about them. I mean, we can talk about your particulars if there's other work that you could do, you know, cut out or something instead in order to accommodate that. We'd have to talk about your particular project. But we cannot go over those caps.
Can the R61 phase of 18761 involve establishing an existing model in-house, not de novo?
So 761 is the therapeutic agent characterization and in vivo efficacy studies. So if you're establishing a new model, then you would probably do better looking at 18763, which is the animal model development.
Next question.
Just to follow up, yes, you can work with an existing model. It's just a matter of if the model is not well validated, although it's already established in-house, your studies you would propose would be how you would go about validating it.
Right, you're talking about 763.
Yes, in 763.
Yep.
Next question. Can these projects involve CROs?
Yep.
For PAR 762, if the HTS involves CRISPy or CRISPA library screen, would that be considered?
Yeah, CRISPR screens would be considered.
Next question. What are common non-science reasons that projects fail to transition?
I don't think we've had anybody not transition for non-science reasons so far. I think, you'll see in your FOA that it's always subject to availability of funds and things. We haven't had that happen yet, but to use my imagination, you might imagine something changed in the field such that there really wasn't need to develop the therapeutic anymore, or something crazy like that. But so far, we haven't had that happen.
Given the degree of non-science efforts and tasks required, can effort for administrative staff be included in DC? How much preliminary data is required?
Okay, I think we've already talked about preliminary data. The rules around budgets for this are the same as RO1s and any other mechanism in terms of what can be direct and indirect costs.
If the amount of work needed for entry into the blueprint requires more than can be covered in one round of R61 and R33, can one apply for additional rounds if the first round is successful?
Right. So, for example, you might imagine somebody needing to do 761 for in vivo efficacy studies, and then also after doing 762 for a high-throughput screen to identify the compounds in the first place. That would be appropriate.
Are there restrictions on how the time and money are divided between R61 and R33 phases?
Yeah. So for the money, you cannot have more than $499 in any given year, and you cannot have more than $750 direct costs for the whole thing. Other than that, you can divide it how you want. For the money, you need to have at least 1 year in R61 and at least 1 year in R33. A lot of people choose to do a 2-year and then 1 year, or a 1 year and then 2 year, rather than have it divided completely in half, only because that saves you from doing that progress report, because you can submit it with the same time you do your R33. But if it makes scientific sense to divide it straight down the middle, you can do that.
Next question. For PAR 18761, if a compound has been identified and is commercially available, can we include studies to make this compound better and unique during the initial phase of the grant?
Yeah, so, a reason to not do something commercially available is, for IP reasons, but if you're going to, develop it into something that you have now IP on, that's fine.
If your work in R33 reveals your lead compounds actually represent a family of new structures that might be better suited, but there are not enough available and they would need to synthesize more.
I'm guessing that should say, is that supportive? So that sounds rather specific. And I'm guessing you're talking about the high-throughput screening, or the screening and target, screening and agent identification, FOA. You can propose a small amount of focused synthesis if you find some whole new family at the end of your grant that you need to develop more of. If you get to that point, let's talk about it, but that would probably be all right. I guess I'm not quite sure what FOA we're talking about or where this is coming from.
Can a small business still apply if this two-phase mechanism fits very well with their project plan?
You are allowed to apply, but again, even if it sounds like a great fit, you can still do all of this in the SBIR General Omnibus, and it's still a better way to go because, like I talked about with the set-aside funds.
Is joint research between an academic lab and a company suitable for this mechanism, or would that be better suited for the SBIR?
Yeah, so If you're eligible for the SBIR or STTR, the STTR is something you should definitely look at if you're doing joint research. If you're eligible for those, I would recommend going with SBIR or STTR. If you're not SBIR or STTR eligible, then we can talk about IGNITE.
And to follow up on that, I do know that the SBIR team is trying to set up a webinar for them in the next couple of weeks. So everyone that is applying who's on this call, I will send that out to them as well.
Great. Thank you, Tim.
In the next question, is a two-institution collaboration between a company and academic center okay for applying for PAR 18761?
Yeah, this sounds a lot like the last one. It's okay. You're eligible. You're allowed to do that. But again, the SBIR or STTR would be a better way to go if you're eligible.
What if the R61 is completed in less than the planned 3 years, in the planned time, especially if it was planned for 2 years? Can you transition to R33 early?
You know, we much more often see the other issue where the R61 is not done in time and people need no-cost extensions. I've actually not had that happen. I'm not sure if that's doable or not.
I would say theoretically it's doable, but that's more of a question for review. So you want to structure your application so that it looks feasible for the time that you're proposing it in.
Mm-hmm.
What's the success rate of getting funded through this mechanism?
You know, I don't know that we have the exact number. So unlike R01s and R21s that are percentiled and we pay by pay line, these are not. So we don't have a, cutoff where X percent get funded. It's really dependent on how many good applications we get, what kind of issues are in the summary statements, are the issues in there things that we feel like are fixable. So we don't really have a, success rate cutoff or a pay line that we go by with these ones.
Okay, so the next question is a previous question that they've reworded. How best to do iterative development? So, for example, if your work in R33 reveals your lead compounds actually represent a family of new structures that might be better suited, but there are not enough available, resources, or there needs to be a new synthesis of this new family in order to continue, how would you address that?
All right, so in general, this sounds like, something comes up in the middle of your research that you didn't plan for and you didn't budget for or have time for. So, and that's larger than, a synthesis question, but that is something that sometimes comes up. So if you need to change scope or rebudget, it's just like any other grant where we would have to talk about it and pre-approve. Things like supplements, again, we don't do them a lot, but if something really weird comes up, we can talk about it. But yeah, like any other grant where something, there's some change of scope or something, we'll talk about that in your, once your grant is going on. Now, you can also plan ahead a little bit and have alternatives and backup plans and things like that, and that's always a good idea when you're writing grants. But I guess it kind of depends on the, the particular situation.
All right. If you have a question, you can go to the front of the line. So we're going to give it about 1 minute to give people a chance to ask any last-minute questions. I guess we can use, while we're waiting for more questions, I just wanted to point out that, and we've already mentioned this a couple times, but there will be a biomarker webinar next Wednesday on the 11th, and I believe that's at 1:30 or 1. It's at 1 o'clock p.m. Eastern Time. And then on the following week, on December 18th at 3:30 Eastern, we will have a blueprint webinar. So we definitely invite you to join both of those.
And if somebody wants information on registering for that, they can email you?
Yes, they can email me.
Okay. And they can join our listserv.
Yes, they can.
And they'll get updates on future webinars.
All right, so it doesn't look like we have any other questions. Okay. So I think we're going to end it. Thank you, everyone, for joining us. Please reach out if you have any suggestions on how we can make these webinars better. Mm-hmm. And like we mentioned earlier, we will send out this PowerPoint to everyone. And the hope, should this recording work, knock on table, we'll be able to send that out to you as well. But definitely expect the PowerPoint presentation sent to you this week.
Yep.
With that, I'd like to say thank you.
Thank you.